Welcome to our presentation. My name is Lindsay Thompson, and I am joined with classmates Mia Johnson and Jason Britt. And today we are going to be discussing food intolerances for the class CSPH 5431. In this slide, we just wanted to list out the different aspects that people researched. Jason will be discussing antecedents. I myself, Lindsay, will be discussing triggers, and Mia will be discussing mediators within this presentation. And we also felt it was important to differentiate between allergies and intolerances before going any further. Allergies are an immunological response by IgE. Typically, this occurs right when someone ingests food. Usually at its most severe form of a reaction, it causes anaphylaxis, so trouble breathing, throat swells up, sometimes causes cardiac issues. As where intolerances are a non-immunological response, this usually does not occur from a one-time ingestion of food like it can with allergies. Intolerances are considered a type 4 cell-mediated delayed hypersensitivity, so it usually takes multiple times of seeing or eating the same food for this response to occur. And so we're going to be discussing the less severe, less acute form food intolerances. And we just wanted to differentiate that because sometimes I think allergies and intolerances, those words get used interchangeably. So we just wanted to make a distinction.
I'm Mia Johnson, and this is a brief introduction to the content of our presentation. We will be presenting information about the antecedents, triggers, and mediators of food intolerance. Antecedents can include genetic, environmental, microbial, geographical, or agricultural indicators that have the potential to predispose individuals to food intolerances such as dairy and gluten, amongst others. In addition to antecedents, we will also discuss triggers that can aggravate or initiate one's symptoms related to food intolerances. According to a literature review conducted by Tuck and colleagues, triggers of food intolerance can be both modifiable, like fermentable oligomonosaccharides and polyols, FODMAPs, wheat, histamine, food additives, and bioactive chemicals, or non-modifiable, like sucrase isomaltase deficiency. The final topic we will cover is mediators that perpetuate and continue a food intolerance. The primary mediator of food intolerance is diet. A mediator that often goes hand in hand with diet is gut health and gut makeup. For example, the enzymes and microbiota present in the gut can perpetuate or help heal food intolerance. The protective barrier inside the intestines and gut can also play a role in mediating food intolerance.
All right, so antecedents to food intolerance. So when looking at food intolerance, I think it's a good question to ask to start off with. Why do we become intolerant to certain foods? Why do we become intolerant to the lactose in dairy or the gluten in grains? And why is it becoming so common in our culture? To look at some stats behind that, 15 to 20% is estimated that of the human population has some type of food sensitivity. And so when you're looking at stats and looking at studies that are that speak about food intolerance and food sensitivities, they use kind of the words interchangeably. So for the purpose of this review, we're just going to be looking at, what do you eat that causes the symptoms of food intolerance, such as gas, bloating, abdominal cramping, and the commonalities amongst those studies are pretty standard in that it's the food components, the macronutrients, along with the toxins that are in food, such as the glyphosate. We'll talk a little bit about that later on in this review, and how that impacts the body. The different components of wheat and gluten, the byproducts of wheat digestion, which are gluten and the peptides, gliadin, and the sensitivities we have to those that cause celiac disease and non-celiac gluten sensitivity, along with enzyme and transport defects, which are microbial based food intolerances, and the makeup of our microbiome and how that impacts how we digest and absorb food. So when looking at food intolerance and why it's become so prevalent in our culture, especially in the westernized culture, it's probably a good idea to ask, what are we eating? What are we eating that is possibly predisposing us to having food intolerances to begin with? And so, in looking at our diet, our typical diet is a standard American diet where a majority of people are eating a lot of processed foods that contain, sugars, salts, oils, added fats, a lot of toxins. That is definitely a contributing factor to food intolerance for sure. That's supported by a national health survey from 2003 to 2006 that showed that only 16.5% of the people in the US are consuming a healthy diet, which is incidentally increased to 32% if moderate exercise is factored in. So there's probably a few lifestyle factors that are contributing to food intolerance that are non-food based. 60% of foods eaten in the U.S. contain added salts, added fats, added solid fats, or added sugars. And the two highest groups, coincidentally enough, are dairy and grains that contain either added fats or added solid fats or added sugars. And oddly enough, those are the two groups that we'll be discussing further in the antecedent as causes or antecedents to creating food intolerances to begin with. So starting with dairy intolerance, or intolerance to lactose, um, is a very, very common food intolerance that is very prevalent in much of our culture. Um, a lactose intolerant is basically due to a lack of or a decrease in the lactase enzyme, which is needed to break down and absorb the lactose found in dairy products, milk, cheese, etc. Three different types of lactase deficiency. um, is primary lactase deficiency, which is a natural decline in lactase that happens after infancy. It's a natural decline that happens in many people, in some cultures less than others, and we'll take a look at that too. Some of the history behind the different demographics and geographic rates of lactase deficiency, primary lactase deficiency. Um, and secondary lactase deficiency, which is gastro and it's health related. So gastrointestinal infection, irritable bowel disease, abdominal surgery, any type of illness that can temporarily cause a temporary decline in the ability for the body to express lactase to digest lactose. Usually after the illness, lactase production returns to normal. And then there's finally congenital lactase deficiency, which is extremely rare, but also extremely fatal. It's where an infant is born without the ability to express lactase, and so they can't digest the mother's milk. And it's usually um, it's found in commonly in Finland, but there's very few cases per capita. So it's just one of those things that can happen, but doesn't often happen. So here's a cool little slide that shows a picture of the different types of a lactose intolerant individual versus a lactose intolerant individual, and I think it's good to have a little visual of how this works. And so on the left here, you can see that a lactose tolerant individual, you have lactose, which is broken down through dairy digestion, normal dairy digestion in the small intestines. And so we have a lactase enzyme that is expressed in the small intestines. It takes the lactose and converts it into glucose and galactose for sugars that the body can utilize. And here on the right, we see lactose coming into the small intestines without the little lactase enzyme to digest it. And when that happens, when there's a lack of lactase, you have the lactose that makes its way into the large intestines. Um, and then you have bacterial fermentation that happens, which then causes the gas and acids to build up and, you know, produces the symptoms of food intolerance, which are flatulence, abdominal pain, different things like that. And as I mentioned, one of the primary causes of lactose intolerance is the inability for the body to create the lactase enzyme. Expressions of the lactase enzyme is mediated by the LCT gene. LCT mutations can occur over time in a culture, and lead to a reduction in lactase production. Or it can be an indicator of lactase persistence. And so a couple of these studies that I looked at both lactose intolerance and the genes that were related to lactase persistence. So what causes LCT mutations? And there's several hypotheses um, that have been postulated. Um, the first one from Holden and Mace in a 1997 study is called the cultural historical hypothesis. And they looked at different aspects of different cultures all across the globe, and they found that pastoralism was, populations that kept livestock and derived a significant number of calories from dairy products, was one of the main indicators that really drove um, where the LCT mutations happened, and whether or not populations were maintained lactose persistence past infancy. Holden and Mace also looked at two other studies, or two other hypotheses: the arid climate and the elevation hypothesis, where they looked at lactose persistence in areas that were drier climates or at higher elevations, to see if that played any role in lactose intolerance. But they really went back to the pastoralism as being one of the primary indicators of whether or not a civilization or culture was lactase maintain that lactase persistence. And another study in 2014 looked at pastoralism in Africa. They found that the lactase persistence genes existed in a high number of LCT SNPs where pastoralism still exists. A lot of modernization has happened in Africa, so there's actually a high amount of lactose intolerance still in Africa, but in regions where they do practice pastoralism, um, they exhibit a lot less lactose intolerance than other areas. And this image is a continuation of the last study mentioned in 2014 in Africa, where they looked at some common SNPs, so which are different regions of the LCT gene that were markers of lactase persistence. Different areas, different pastoralist areas, had different SNPs compared to non-pastoralist areas, and so different prevalences of lactase persistence exists all throughout Africa. Um, as I mentioned, on the whole, Africa experiences a high level of lactose intolerance, but they still have areas where there's very little lactose intolerance. So there seem to be a lot of genetic, but also environmental factors um, that play into whether or not we're able to digest lactose. They had some areas that they found where people didn't have these markers for lactase persistence, but yet they were still able to digest lactose, which really signifies that there's some other environmental or possibly microbial elements to our ability to digest lactose. So this little image shows pastoralism over the past 10,000 years, and this is supposed to be an animated GIF, but it isn't quite working right now. Um, but if it was working, you'd see that a countdown from 10,000 down to present day, that we started pastoralism in northern Africa here, and then kind of migrated to northern Europe, and then into Asia, and then in the past couple thousand years over into the Americas. And this is really just to show that as we migrated in different areas of the world and have had pastoralism present for different lengths of time, and subsequently, one of the main it's subsequently been one of the main selective pressures that have been driving genetic shifts over time. And so, continuing on the pastoralism thread, this is some of the more of the history on pastoralism and some of the impacts on human health. From Nutrition and Physical Generation, a book by Weston A. Price, should be familiar to some of you listening. He did some tests on butter. From 1928 to 1938, there were some studies done on butter samples that he collected every few weeks from different areas throughout the US, Canada, Brazil, Australia, New Zealand. Um, these samples were analyzed for vitamin A content and also unknown element at the time, which he called activator X. And which is now commonly be thought to be vitamin K2 and MK4, a type of vitamin K2 that's produced by animals. Uh, and Price identified in the study that pastoralism and what type of fodder we fed the animals in these areas really had a direct correlation to the vitamin content in butter. So he noted vitamin A and vitamin this activator X as being as major indicators of health and wellness in these areas. And so two of the pathologies he looked at were heart disease and pneumonia mortality in these areas. And he found that samples in those areas contained varying levels of vitamin A and vitamin X, and there was a direct correlation between human health and the concentration of those vitamins. And he also found deformities in animals in those areas. So he found like deformities in chickens and turkeys and how they were raised, and what kind of deformities these animals had. Um, so as I mentioned, activator X now thought to be vitamin K. We know now that vitamin K plays an important role in heart health and bone growth, um, which I'm sure Weston A. Price probably knew over about 100 years ago when he first started doing those studies and started seeing those pathologies develop. Um, and we know that the makeup of the microbiome is mediated by vitamin K. So um, the microbiome is directly involved with vitamin K as well, and so that's one of the major factors as to maybe some of the pathologies that are occurring that he noticed with activator X in his studies. So continuing further with vitamin K in the microbiome, some elements of vitamin K are produced by the gut microbiota, which I found was really interesting. That up to 50% of our vitamin K2 is not dietary; it's actually produced by the gut microbiota itself, and that was from a study by Hill in 1997. Uh, and that also in this Ellis study from 2021 this year, it was found vitamin K deficiency alters the gut microbial community composition, which I also found really interesting that um, a vitamin could have such big impact on the microbiome itself. So we know that changes in the gut composition, which Ellis noted in the study, um, can contribute to symptoms of food intolerance. So, changes in the microbiome, can contribute to SIBO, small bowel intestinal bacterial overgrowth, and FODMAP digestion issues. And obviously, lactose is a FODMAP, so Lactobacillus acidophilus, I think that's right, aids in the digestion of lactose. So that could be maybe one of what we saw in Africa, where some people didn't have um, the SNPs that accounted for lactose persistent, but were still able to digest lactose. So, in looking further at some of the antecedents to food intolerance, I thought grain and gluten intolerance would be another good one to take a deep look at, just because it's so prevalent. Um, and there might be some overlap there between dairy and gluten intolerance, since a lot of people seem to exhibit both intolerances at once. Uh, Gluten is a protein found in wheat, and wheat's digested into gluten, which is further digested into peptides and amino acids. Gliadin, which is found to be one of the main triggers of gluten intolerance, is one of the peptides that gluten is digested into. Undigested peptides lead to symptoms like gas, bloating, abdominal pain. The different types of gluten intolerance that we know of, that we diagnose, are celiac disease, non-celiac gluten sensitivity, and wheat allergy. And each one of these really varies from study to study. Um, some studies say there's 1% of the population, Western population, has celiac disease, and um, or some studies say up to 6%. So it depends on what study you read, but it seems to be pretty low prevalence, but um, really gaining, probably underreported, um, based on a Gallup poll from 2015 that showed up to 20% of Americans opt for a gluten-free diet, which really shows that people may be at differing degrees of grain and gluten intolerance, and maybe a lot of it going undiagnosed. But it seems like, yeah, it's probably rising in a lot of different ways. So continuing along the thread of why gluten intolerance is on the rise, um, there's a study in 2009 that looked at frozen blood sera samples from 1948 to 1954 when compared to samples taken in 2009, and it showed a 4 times increase in celiac disease, which I thought was really interesting. That there was such a drastic rise, and from the 50s all the way till now. And another study in 2020, study on celiac disease, showed that it has been increasing 7.5% a year each year for the past several decades. So it seems to be quite a bit, quite on the rise, quite a bit. So the question is, why are we becoming more progressively predisposed to gluten intolerance? So one of the main hypotheses on why we might be becoming more grain and gluten intolerant is that we lack the digestive gut microbiota to be able to do that. And so, in looking at some reasons why we might be experiencing shifts in the microbiome, MIT researcher Dr. Stephanie Seneff, in her new book Toxic Legacy, and several articles that she's written, um, really puts forth the theory that it's the way we do agriculture and wheat farming now that is causing a lot of driving a lot of the gluten intolerance. Glyphosate, one of the main ingredients used in the common herbicide Roundup, is also a patented antibiotic, and it's used heavily as a wheat desiccant, which means it's sprayed on wheat crops to dry out before they get harvested. So anytime we consume non-organic wheat, which is a lot of the standard American diet is a lot of wheat-containing products that are non-organic. We're getting a heavy dose of this pesticide, also an antibiotic, along with that. So that seems to be a likely factor as to what might be causing some of this gut dysbiosis that we see in SIBO and different things with digesting FODMAPs. And so, looking at a couple of these bacteria, um, there's one bacteria type, lactobacillus, that is sensitive to glyphosate. So it's been shown in vitro to be sensitive to glyphosate, and that's one of the main bacteria that helps us detoxify the gluten peptides, the gliadin, um, that makes it easy to digest and able to digest. Whereas the Pseudomonas are actually glyphosate resistant, and they create immunogenic peptides or inflammation, um, which could be one of the main reasons that we're experiencing so much gluten intolerance. Okay, so continuing along with the Dr. Seneff's hypothesis that glyphosate could be driving a lot of what we're seeing with celiac and non-gluten sensitivity. Um, the little image on the left here shows glyphosate usage and how it's progressed over time. And so in the early 90s, we were not using that much. It was mostly used as a residential weed killer. And as we progressed over the past 30 years, we've been using it more and more progressively, and not only wheat farming, but just conventional farming in general. So it's much, much more prevalent than it was even 30 years ago. And oddly enough, 30 years ago is when we started seeing, here's the early 90s, right here in the middle, is when we started seeing like a sharp incline in celiac and non-gluten sensitivity. Um, so it's been around for a while, but it really hasn't taken off until like the past 30 years, where it's just become really a prevalent issue in our culture. So it seems like it's pretty good circumstantial evidence. Correlation doesn't equal causation, but this is pretty good circumstantial evidence that there's at least something going on with chemical usage and changes and shifts in our microbiome that might be contributing to the lack of ability to digest gluten. And so here we just have a little slide that shows um, kind of a cartoony image of what someone with celiac disease might experience in their gut compared to someone with no active disease. Um, and here on the left, we have uh, uh, Pseudomonas um, organisa. I'm pretty sure I didn't get that right, but It's the Pseudomonas bacteria that are the ones that cause the immunogenic peptides, which trigger the immune response, compared to um, the image on the right here, where it shows the lactobacillus, um, digesting the peptide, the gluten peptides, in the gliadin, and making bacterial detoxified peptides, which makes them digestible and not trigger the immune response. So in conclusion, antecedents to food intolerance, we know that genetics plays a major role in the predisposition to certain food intolerances, such as the LCT gene mutations that cause us to be lactose intolerant and not express lactase. Microbial composition and subsequently gut dysbiosis, we know plays a major role in, um, or likely plays a major role in the digestion of gluten peptides, and the prevalence of celiac and non-celiac gluten sensitivity. And we know that microbiome plays an endogenous production plays a role in endogenous production of vitamins such as vitamin K, which also have an impact on the microbiome, and that there are likely geographical, demographic, environmental factors, all playing various roles, that are either contributing or the cause directly of food intolerance. The genetic microbial shifts that happen over time and can predispose various individuals and populations to food intolerance. Uh, Pastoralism is shown to be a key factor that creates a selective pressure that facilitates genetic shifts over time.
And now we're going to transition from talking about antecedents to triggers. And when we're conceptualizing triggers, the question that we're trying to answer is what initiates the food intolerance response? Tuck and his colleagues did a very comprehensive literature review in 2019 and kind of categorized these triggers into 5 main categories. I will be discussing these 5 categories and noting some proposed theories or mechanism of actions that relate to these triggers. The first one that we will discuss are FODMAPs, which are fermentable oligodimonosaccharides and polyols. These are kind of a large group of short chain carbohydrates, and there's 2 kind of proposed mechanism of actions as to how FODMAPs may initiate this food intolerance response. The first is that these short chain carbohydrates have an osmotic effect, meaning that they bring water into the lumen, thereby increasing the volume of the intestines, which can then cause some discomfort. Another theory that is proposed is that if these chains get into the colon, bacteria then feed off of these chains and produce excess gas, which can cause bloating and some other discomfort. Next is wheat. When discussing wheat as a trigger, the main component associated with food intolerance is typically gluten. So this component can be found in a wide variety of foods that are made with wheat. Most likely you have already eaten something today that contains this. The exact mechanism behind this trigger is not really known, but there are a few proposed theories, and they include kind of the following. Increased lipopolysaccharide binding protein, which this is a component that then is associated with an immune response, which then is associated with an inflammation response, which is where some of the symptomology can come up with for food intolerance. Increased eosinophils, innate and adaptive immune activation, increased intestinal permeability, and changes within the gut microbiota. And finally, histamine. This is a type of amine that can be present in foods, particularly in fermented and canned foods. And when someone accumulates a large amount of histamine, either through ingesting a bunch by eating these foods, your body produces histamine naturally, but it's usually not enough for it to cause any issue. Your body does a really good job of regulating this on its own. But if you do eat large amounts of histamine and you're deficient in something called diamine oxidase, which is an enzyme that breaks down this ingested histamine, this can cause a variety of symptoms, particularly within a lot of bodily systems: respiratory, gastrointestinal, neurological. And if you think about it, histamine is the amine that is associated with kind of seasonal allergies. So if you have ingested a lot of these types of food, your symptoms are going to look more like seasonal allergies. So you could have things like headaches, abdominal pain, runny nose, itchy skin, etc. The fourth category is food additives and bioactive chemicals. These are things that are found in a large number of processed foods. They're commonly added to foods and identified by the different functions that they have. So these things can be used to thicken up a food, make food more cohesive, preserve a food, or even enhance its flavor. And currently, there's kind of a lack in research, particularly with controlled trials related to these different types of additives and chemicals, in determining the mechanism of action behind how these additives relate to its effect in food intolerances. However, I did find a study that was conducted by Malaise and colleagues in 2020 where they had mice ingest various bisphenols, so BPA, BPS, and BPF, to determine if this influenced whether these mice would develop food intolerances, and they measured this through immunological response. And the researchers found that even just low dose ingestion of all of these bisphenols increased both their humoral and cellular mediated responses. And that essentially translated into their decreased ability to tolerate various oral dietary antigens, which is just a very fancy way of saying they ended up developing some food intolerances as a result of this low dose ingestion of the bisphenols. And the final category is sucrase isomaltase deficiency, which causes the inability to break down sucrose and starch. This is an example of a non-modifiable trigger because it's due to either a genetic predisposition or sometimes it occurs through an event that has happened to someone, although most time it is a genetic predisposition. And these types of triggers are commonly related to a deficiency in some form of a particular enzyme that helps break down certain substances, which then causes a buildup of these substances and ultimately leads to damage in the intestine. So in the case of this particular enzyme, sucrase isomaltase, the inability to break down sucrose or starch allows these substances to show up further in the intestines, which ultimately results in symptoms such as diarrhea, abdominal pain, and or bloating. And while there are many more potential triggers that could be related to food intolerance, these are sort of the 5 main categories of triggers that have been identified in literature as the most commonly researched causes to date.
What we eat will always be the biggest contributor to the continued presence of a food intolerance. For example, consuming foods containing the triggers previously mentioned by Lindsay will always perpetuate symptoms of the food intolerance related to those triggers. One way that we can gain a more acute understanding of how diet is such a crucial mediator is by examining one of the leading intervention plans for food intolerance, an elimination diet. An elimination diet works through a structured plan of removing and reintroducing foods to the diet. An elimination diet is most commonly used to diagnose or recognize a food intolerance and can be continued as a means of preventing symptoms. Patients utilize this diet by abstaining from all foods that seem to be causing the symptoms of intolerance for around 2 weeks. A patient will, at that time, typically begin a diet of foods that very rarely cause a reaction. Once they have created a neutral based diet that causes no symptoms of intolerance, they slowly reintroduce foods that have been eliminated one at a time. And any reintroduced food that causes significant symptoms to appear is identified essentially as an allergen for the person. This diet is notably only a truly viable option when these foods do not pose risk of anaphylaxis reaction. The notion of an elimination diet paints a clear picture of how diet is a primary mediator in food intolerance. Continuing to eat particular foods will perpetuate the issue of a food intolerance, while choosing to alter one's diet around the food items causing the symptoms of intolerance will aid in helping the issue of food intolerance. In addition to diet, the makeup and health of one's gut can be a crucial mediator in whether or not one's food intolerance continues. Diet can work in tandem with characteristics of our gut health to create a healthy environment that begins to heal symptoms of food intolerance. One aspect of gut health is the possession of certain enzymes that help in the processing of food. Whether or not one's gut has a particular enzyme can be important in understanding whether or not a food intolerance will continue. As mentioned in the antecedents portion of the presentation, lactose intolerance is one of the food intolerances dependent on the presence of a particular enzyme. In this case, it is lactase to process lactose. Lactase activity occurs within the small intestine. This is a clear example of diet and the biological makeup of one's gut coinciding as mediators of food intolerance, because while the consumption of lactose is perpetuating the intolerance, so too is the lack of lactase activity within the gut. Another aspect of gut health that can be more readily changed by a person is the microbiome of their gut. This coincides largely with diet, and again, we can begin to analyze this mediator through examining interventions. A specific focused diet, such as a specific carbohydrate diet or a low FODMAP diet, can illustrate how diet and gut health work together as mediators. Due to the immense amount of genes within the microbiota of the gut, anything influencing the microbiota can affect gene expression and lead to symptoms and conditions of intolerance related to food consumption. The two diets, low FODMAP and specific carbohydrate diet, are similar to an elimination diet. However, they focus on removing very specific foods from the diet, centered around the idea of creating a healthy gut microbiome. Both of these diets, in slightly different ways, focus on the idea of reducing poorly absorbed and highly fermentable carbohydrates, with the idea in mind that due to the way these carbohydrates are digested, they have the potential to cause fermentation and overgrowth of bacteria and yeasts inside the gut, which will then switch the microbiome toward a pro-inflammatory profile. Usually, the structure of these diets occurs strictly only in times of flare-up of major conditions or symptoms. But when the body is in a less reactive state, these particular carbohydrates can be eaten in moderation rather than eliminated. It is this differentiation that illustrates how both diet and gut health can be mediators. The food specifically is not the only mediator because it can be eaten in moderation without causing significant symptoms of intolerance. However, for certain individuals, if consumed frequently enough or built up over time, it can actually shift the microbiota in the gut, eventually causing the individual to experience symptoms of food intolerance or conditions associated with food intolerance, such as inflammatory bowel disease or irritable bowel syndrome. Lactase activity and microbiota are specific factors within the gut that can mediate food intolerance. But the general structures of the gut itself are equally important in protecting against food intolerance. For example, as the immune system is present in mucosal membranes, the protective barrier of the small intestine is crucial in maintaining a healthy gut and overall body health. It is important that our intestinal lining's protective barrier stays intact. When this barrier is destroyed, we are at risk to experience intolerance of certain foods. Our normal intestinal barrier allows for the absorption of beneficial dietary elements. It also aids in preventing harmful substances, particularly pathogens, from passing through the intestinal barrier. But if there is any disruption or gap in this barrier, we can have pathological states in the body. In children, there are already bigger gaps within the intestinal lining that allow for immunoglobulin incorporation, particularly from breast milk. However, this protective barrier can also have gaps created in it in older age through damage caused by diet or toxins, especially a hyperacidic diet. When these gaps are present, whether it be biologically, such as in children, or caused by a particular diet, it can not only allow pathogens to pass through the barrier, but it can also allow other molecules like food proteins, such as casein or gluten, to pass through the barrier and be mistaken as pathogens, therefore initiating an overhaul immune response. This internal miscommunication, where a protein found in food is misinterpreted as a pathogen, creates a learned immune reaction that causes a person to eventually develop a condition where they're unable to tolerate a particular food. So not only does diet play a role in which protein makes its way through a damaged barrier, but poor diet can also be a primary cause of the damaged intestinal barrier itself. So, similar to enzyme production and the microbiome, the relationship of the gastrointestinal barrier illustrates both diet and the biological makeup of the gut as mediators in food intolerance. Throughout the discussion of antecedents, triggers, and mediators, multiple potential interventions have been mentioned to prevent and heal food intolerance. One example is the option of a complete avoidance of triggers. A patient could examine any of the triggers previously mentioned in the slides and work methods into their lifestyle of avoiding these particular triggers. There is also the potential, as mentioned in the mediator section, to follow a more specific diet, such as a low to no FODMAP diet or a specific carbohydrate diet. And this could be used to either preventatively avoid triggers, could be used to avoid foods that one seems to be having a reaction to, or these diets could be used to create a healthy gut microbiome that could um, retroactively heal some food intolerances and more often prevent food intolerance. And the last, most commonly used intervention and often thought to be the most effective as of now is the elimination diet, as mentioned in the mediator section. This diet is a structure of removing foods from your diet and slowly reintroducing them one by one until a patient recognizes a food that seems to be causing a symptom of intolerance within them, and then they can choose to remove that food from their diet. It can be an easy way to diagnose. However, there is currently no gold standard of a method of diagnosing food intolerance because these symptoms that occur when a food is introduced or food is consumed are very different and present very differently between individuals. They can include bloating, diarrhea, abdominal pain, dizziness, headaches, and vary greatly from person to person. They also tend to be common symptoms of other conditions, and it can be hard to link them specifically to a food intolerance. There are also multiple potential causes of a food intolerance, as mentioned throughout our entire presentation. There can also be multiple causes linked to some of these symptoms. So, again, it can be difficult to identify specifically a food intolerance as the initiator of some of these symptoms.
Okay, we've looked at some antecedents, triggers, and mediators to food intolerance, and now let's look at some solutions. As Mia mentioned, low FODMAP or no FODMAP or elimination diet are great strategies to use when trying to diagnose what we might be intolerant to and to help mitigate the symptoms of food intolerance to certain foods. But there's also some overall great strategies to use in general to keep our bodies healthy and digesting well. Eating local and fresh is a big one. Anytime we're eating from farmers markets, local CSAs, or co-ops, chances are we're eating some good quality, nutrient-dense foods, which can only help our overall health and digestion. Limiting sad foods and processed foods with added fats, oils, and sugars. High starch and sugar diet is shown to cause overgrowth and things like SIBO, bacterial overgrowth, and things like SIBO and overgrowth of Candida and Klebsiella, which are inflammation-causing bacteria, and which also might be playing a factor in food intolerance. Organic foods to avoid toxins, like I mentioned. Non-organics contain glyphosate, which is shown to have a major impact on the gut microbiome and reduces the bacteria that can help digest gluten and lactose. Eating a wide variety of foods and moderate amount of fermented foods. Both of these things will help build gut diversity and help digestion and help mitigate the symptoms of food intolerance. And things like exercise, sleep, and stress. I think sometimes we forget about food intolerance. The rest of our body plays a role in that as well. So these lifestyle factors um, are going to play a role in our overall health and wellness. And there's even some studies out there that show that things like stress and exercise can change the makeup of your microbiome. So really, just about everything we do impacts the microbiome, which either predisposes us to having food intolerance or having triggers or mediators impact food intolerance as well. So I'll leave you guys with a quote. Let thy food be thy medicine, and the medicine be thy food, from the father of modern medicine, Hippocrates, in about 500 BC. So I think that's a good strategy to leave you guys off with. And thank you for listening to our presentation. Our references are in the final two slides, and we look forward to connecting with everybody and our fellow cohorts in the forums. And please let us know if you have any questions. Thanks.